Hi, my name is Dr. Joseph Gleeson from the University of California, San Diego, and I'm very excited today to talk to you about our new paper entitled Developmental and Temporal Characteristics of Clonal Sperm Mosaicism. De novo mutations are an important cause of disease and an important contributor to human evolution. De novo mutations start from the germ cells from the father or the mother that contribute to the zygote. And when a mutation is present in one of the germ cells, it shows up as a new mutation in the child and will often be missed in the sample from the parents. So it looks like it's a de novo mutation, but it's actually originating as a clonal mosaic mutation in one of the parents. And it's been known for a long time that the number of de novo mutations correlates very closely with the age of the father at conception, from work from Kari Stephenson and Jonathan Sabat from a decade ago. The number of de novo mutations doubles about every 14 additional years of the father at conception. And so in this project, we're dealing with clonal sperm mosaicism. It's one of the two major types of mosaicism. In sperm, clonal sperm mosaicism originates when the father was an embryo. And then after the first several cell cycles, the primordial germ cells are formed. Mutations that happen early in this time are going to be clonal. Mutations that happen late in either spermatogonial stem cells or later in spermatocytes leading to sperm will show up as non-clonal, and both of these make important contributions to de novo mutations and ultimately disease in children. We asked a couple of key questions for this project. First, how common is clonal sperm mosaicism and what is the variation between men? Two, is clonal sperm mosaicism stable over time as men age? And three, what is the effect of clonal sperm mosaicism on the risk of disease in offspring? I'll be introducing Martin and Xiaoxu to take you through some of the data.
Hello, my name is Dr. Martin Breuss, and I am one of the first authors of this manuscript. So we really wanted to address these questions by assembling a control cohort to study clonal sperm mosaicism. We started by collecting 12 individuals of young age, or YA, between the ages of 18 and 22 years. And this was to find a baseline of sperm and blood mosaicism before age will modify the mosaic variants. We also collected for several of these multiple ejaculates to really understand whether sperm mosaic variants will change over time within one individual. To complement this, we also collected an advanced age cohort with individuals above 48 years of age. And here we wanted to see whether the life experiences and aging will influence sperm and blood mosaic variants. For each of them, we had matched blood sperm pairs, and we also had repeated samples, as I mentioned. And importantly, for all of them, we performed 300x whole genome sequencing to really understand mosaic variants at a very deep and sensitive level.
Hello, I'm Dr. Xiaoxu Yang. So I'm also the co-first author of this paper. By looking at all those data, we found that on average, men have around 30 clonal sperm mosaic mutations. And by looking at different ejaculates from the same individual within 6 to 12 months, we found that allele fractions are well correlated between each of the time points, whereas the correlation between the sperm mutation and the blood mutation are not so well correlated. This also tells us the sperm clonal mosaicisms are stable in terms of their allele fractions, both for the absolute value and also for the relative values. By looking at the YA and AA cohort, we found that in contrast with blood, we're looking at different ages with the number of mutations we found. We see a significant increase of the number of mosaic variants in age cohorts compared with young cohorts. When we look at the sperm mosaicism, the green and the brown shows the sperm-specific and the shared variants are stable in terms of their numbers and also allele fractions over decades of time.
As a consequence of this stability, these clonal sperm mosaic variants have a stable contribution to the mutational load of each offspring independent of the age of the father. In contrast, the non-clonal sperm mosaic variants will increase with age, and you can see here in green the stability of the clonal mosaic variants and the increase in gray for the non-clonal mosaic variants. The clonal mosaic variants will have a decreased relative contribution, here shown in this red line, as fathers age.
We profiled mutations that we detected in sperm for their predicted pathogenicity in offspring, and we calculated that approximately 1 in 15 men will carry a deleterious mutation that's clonal in their sperm, and that has the potential to give rise to disease in an offspring or potentially a spontaneous abortion. And those mutations are present in approximately 5% of the sperm, and with that information we can calculate the risk that any man will have of having a child with a condition due to clonal sperm mosaicism. What we found is that the vast majority of men have almost a 0% chance of risk from clonal sperm mosaicism leading to disease in their children, whereas there are some men that have an appreciable risk. These mutations can lead to congenital disease or miscarriage, and we think that assessing clonal sperm mosaicism in men could potentially reduce the burden of disease from de novo mutations. This work is published recently in a paper entitled Developmental and Temporal Characteristics of Clonal Sperm Mosaicism, and we want to acknowledge all the contributions from the co-authors and their various institutional affiliations. The work was supported by UCSD, Safari, and Rady Children's Institute for Genomic Medicine. Thank you very much for your attention.